All right. Thank you all for coming. Yeah. Thanks, everyone. Super excited to share batch 5 of IndieBio with you. It's amazing that it's only 2.5, 3 years since we started this. I want to thank all of you guys for your time on a Thursday, lovely afternoon. I also want to thank, and I'm very proud of SOSV, the Accelerator VC that we are a part of. And I also want to thank TechCrunch for live streaming our demo day and continuing to be a great partner. Also, I know most of the audience is investors, and so we've set up a website for you called at demoday.indiebio.co that you can go to at any moment to connect with any one of the founders that you see. And so I know that there can be lines and meeting the teams can sometimes be difficult, but this is a really easy way to shoot an email and connect and learn a little bit more about the team. I'd also like to introduce our team, which continues to grow and do amazing things. So June, our science director, Ron, our chief science officer, Ryan, our program director, Alex, our program manager, and our newest addition is Parikshit, who is our analyst. So I just want to take a moment to look back at this experiment we started 3 years ago that we call IndieBio. And it was an experiment around one central hypothesis. Can you help scientists become entrepreneurs? Because we know there's a lot of scientists out there with great ideas and really, really great technology, but helping them turn that into product that can be used to create value was a big question. Can you improve that efficiency? And so it's clear from our portfolio of over 65 companies in the last 3 years that we've learned a lot. And we've seen a lot of failures and we've seen a lot of successes. But the overall trend can point to a success. The market capitalization of all of the companies we funded is approaching $500 million. Total amount of money that the companies that we funded have raised is well over $100 million now. And actually the total revenues reported by the companies for this year are approaching $24 million. So these are real hard numbers that are pointing to both operational and financing success from not just SOSV, but our entire syndicate of investors. To dive into the companies a little bit more, I'd like to hand the mic to June.
Thank you, Arvind. So at the core, we believe in founder-driven companies, and our founders have come from all over the world, over 29 different countries. So they bring a global perspective, and so not just solving problems for Silicon Valley, but to scale their solutions to the rest of the world. Our companies have created over 300 jobs, and now they're bringing their expertise back to their home ecosystems and encouraging others to start companies as well. And I'm super excited to say that 42% of our companies have at least one female founder, which is and this is over 2 times the global average. So IndieBio will continue to strive for diversity for our scientific entrepreneurs. I'll hand over the mic to Ron and Ryan, who will talk about our alumni.
Thanks, June. So I think Ryan and I would like to put some faces on to the statistics that Arvind gave a few minutes ago and talk about some of the companies that are a little more advanced in our portfolio and what's going on with them. What we see is that the companies, as they age, they continue to make progress commercially in terms of their technology and in terms of their attraction and value as investments. So the first story is Memphis Meats. They stood on a stage very much like this 2 years ago at the demo day, and they had presented their first pork meatball that they made for about $3,000. Now they've had tastings where they've produced chicken breast, duck breast, and they've reported a continued drop of the cost of their meat, another threefold in just 2 years. And with that kind of traction, they've been able to attract a $17 million A round this summer with an incredible list of investors, including Richard Branson, Bill Gates, and Jack Welch, right? So we're really super excited about them, and food continues to move forward with us. You'll see today. Ryan?
Thank you, Ron. Also delighted to give you an overview of one of the technical achievements of one of our teams. So Ash, the CEO of Konaku, unveiled this month over at TED Global, so the video is coming soon, the first Konaku core. So Konaku melds biological neurons with silicon to do both sensing and computation. And so we're looking forward to the actual release of the product, but he has been able to, since IndieBio, has been able to both raise his successful seed round as well as large deals with large corporations that are looking to do both sensing and computation with next generation biological chips. I also wanted to highlight Synthex. So we often talk about turning scientists into entrepreneurs. This is an example of two PhD scientists from the University of Toronto that had an idea, which was focused on evolving new peptide therapeutics to treat diseases like cancer. They have since IndieBio raised $6 million. They have developed several new peptides, but one in particular to treat pancreatic cancer. That particular peptide appears to be both highly effective, but also may have uncovered an entirely new mechanism of cell death within cells, which is a huge breakthrough. So this is something that we want to continue and really highlight. So previously unachievable area, which was young scientists starting their own therapeutics companies, is now something that's very real and happening at scale. And we're delighted to be part of that. And so now I'd like to actually hand over to Alex to give you guys an overview of Batch 5 and some of the areas they're involved in.
Great, thank you, Ryan. So we're really excited today to present Batch 5 to you guys. We think this is probably our most scientifically ambitious batch we've ever had. And there's 12 really amazing teams that you're going to be seeing here in a minute. And they're actually, it's a really diverse group, which we're really excited about as well. And we have teams in the future of food and agriculture, industrial biology, therapeutics, like Ryan mentioned. We have healthcare IT, a medical research tool, and we're really excited to be opening a new vertical in regenerative medicine. We have 2 companies working there. Without further ado, I want to hand the stage over to our founders in Batch 5. Thank you.
Good afternoon, everyone. I'm Melanie Mathieu, co-founder and CEO of Finless Biologics, where we are building life with light. What is life made of? Life is made up of interactions, the joy, the cherished memories we all have. At the molecular and cellular level, life is also made up of interactions. What you're seeing here on the screen are cell-cell interactions inside an organ. Cell-cell communication pathways and interactions are necessary for an organ to function properly. When organ failure occurs, it's because tissues accumulate damage over time, disrupting these cell-cell interactions and disrupting these pathways. There's a decrease in organ function, progressive function loss, and eventually complete loss of life. In my research career, I studied these cell-cell interactions. And one day, when I was in the lab analyzing the exact same interactions you see here on the screen, I realized that we could take our high-power, ultra-fine resolution laser technology that we use to image these and reverse engineer it to print the tissue structures around these cells, building a tissue from the inside out. And what was most important was that this super-fine laser could recreate tiny structures, the microvasculature and capillaries that feed organs and exchange oxygen. Without this, you can't print organs. You can't print anything thicker than a sheet of paper. And this is critically important for the over 90 million people in the US alone who are suffering from progressive organ failure. Currently, the market for human organ replacement and tissues is stalled around $500 million a year annually. This consists mostly of non-living tissue replacement and tissue in a dish that's used for therapeutic screening. By printing microvasculature, we believe we can open up a multi-trillion-dollar vertical and provide real human tissue to patients who are suffering. And so with this idea in hand, I designed a laser system that could print microvasculature and then lay down tissue and other cells around it, building a full organ from the inside out. Here at IndieBio, we turn this idea into reality. On your left here, you see our laser-based printing system. It's not projecting a single fine laser beam, but a hologram. That's why the beam is so spread out. And it's projecting it into our tissue dish here on your right, where we are actually printing a structure. But can we print the microvasculature with this? What you're seeing now is a video of printing a human microvasculature structure. The center pore is less than 10 microns across, matching the average size of a human blood vessel or capillary. Around it, in our biocompatible cell matrix, we are printing a ring to trap cells that will grow and differentiate, strengthening that vascular wall. 5 days later, our cells are still inside this microvasculature structure. They're alive, they're healthy, they're dividing. And the dye that we printed with is still contained inside of these blood vessels, or these blood vessel-like structures, speaking to the integrity of the structures we're printing. But how do you build a whole organ from this? That starts with data. The kidney you're seeing here on the left is the kidney structure you're seeing on the left is built of entirely microvasculature structure lit up by India Inc. This data is provided by our partner 3Scan, and we can take this data, run it through our software system, isolate the blood vessels and structures we want to print, and feed it into our laser printing system, recreating native tissue and vascular structures. So do the cells survive in this matrix that we're creating? To test that, we took immune cells. We got them from a single blood donation, we grew them up, we differentiated them, and then we printed them into immune structures. To see if these would work, we took these immune cells and we challenged them with the Zika virus, much the same way you would give a human a vaccine. And within a matter of weeks, we had several human antibody hits that were reactive to the Zika virus. Ultimately, we are a human organ and tissue printing company, and we plan to provide an alternative to organ donation. How will that work? When a patient gets sick, they're sent to a transplant center, and more often than not, they're told they're not a candidate for organ donation. The lucky few that get put on a wait list still have years of time ahead of them waiting for an organ. In fact, most people never get one. 22 people a day die while on the organ transplant waiting list. Here at Prellis, we plan to interface directly with the transplant centers and provide human organs within 4 months of patient registration. How will we do that? Transplant surgeons will send us a tissue biopsy, where we will grow and expand the cells from that until we have enough to print a full organ. We'll run that organ through a series of functional tests, send it back to the transplant surgeon for transplantation into the same patient who donated their own cells. We will give people their own organs back. Our proof of concept is this immune system organoid. We can take this to market by producing human antibodies immediately and running drug screens and tests with them. The first transplantable tissue that we will take to market are human islets. They're the functional unit of a pancreas that type 1 diabetics lose, making them insulin dependent. They have to take their daily shots. There's no good source for islet transplantation. However, it does work to get people off of their daily insulin shots. So we plan to become a provider of islets. Ultimately, in about 6 years, we plan to provide full human organs for transplant. Our FDA strategy around taking islets to market will be the common path. We will start with animal testing early next year, followed closely by new drug indication, and then we believe we can start our phase 1 clinical trials in 2021. We have 3 provisional patents filed around this technology: high-resolution laser-based printing of 3D tissues, printing of cells and cell-containing devices and materials, and synthetic organoid printing. I met my co-founder, Dr. Noel Mullen, at UCSF, and together we have nearly 30 years of combined experience in laser imaging, immunology, and stem cell biology. We have a fantastic team of scientific advisors with deep experience in everything from optical engineering to tissue engineering. We believe there's a bright future for human tissue engineering because we are solving the last hurdle, the final problem, and that is the creation of microvasculature. We're currently hiring for scientists and optical engineers. We closed our seed round early this summer, and we'll be opening our Series A soon. Thank you very much. We're Apellis Biologics, and we're building life with light.
Hello, everyone. My name is Matias Mushnik. I am the CEO and founder of Notco. Notco is a food tech company combining machine learning technology with food science to change the way food is made. See, we humans love food. We eat what we eat because it makes us happy. And we love it because it's tasty, it feels good, it's convenient, it's easy, it's part of our culture. The problem is that most of the food that we love to eat, it's actually garbage. See, food R&D has been working for the last 30 years to maximize the most addictive food possible at the cheapest price possible using the worst ingredients possible. And no wonder the pinnacle of R&D in the last 15 years has been a freaking Dorito. It's true, it's tasty, it's cheap, it's profitable. So we've surrounded ourselves by tasty, cheap, profitable products that we don't need are actually garbage, but we still buy them because often we think that's the only possible way of doing food. 2 years ago, with a team of scientists, we decided to group up and disrupt food R&D and change the way we made food. We needed to make good food, taste good, and to be affordable as well. And we've chosen plant-based, mainly because of 2 reasons. One, because science suggests it's the most efficient and healthier way of sourcing our food. And 2, because there's a specific group of people called millennials have for themselves created a more than $15 billion food market, a plant-based food market. This is the same group of people that will be driving the more than $3 trillion food demand in the next 10 years. So if we already love the food that we're eating, let's not change it. Let's change the way we make the food that we love to eat. With this concept, we develop a technology. First, we take an animal-based protein food, such as milk. We deconstruct it molecularly, so we have a molecular formulation that describes milk taste, texture, smell, and color. We do the same with plant-based ingredients. And then we've developed a machine learning algorithm called Giuseppe that is helping us predict which combination of plant-based ingredients should result in the same molecular structure as the animal-based protein food, such as milk. The algorithm's output are recipes of plant-based ingredients that, when combined together, they should result in something really, really close in taste, texture, smell, and color as milk. All of the sensorial data coming out of these formulations is then uploaded as feedback as the algorithm. So the algorithm is basically training to understand the links between the molecular components in food and the human perception of taste and texture. Then every time we iterate, the end result is a plant-based formulation that should taste really, really close to the actual product. The end result, a mayo that tastes like mayo, but without the egg. A yogurt that tastes like yogurt, but without the milk. A milk that tastes like milk, but without the cow. And I'm very happy to display this. This is our first product in supermarkets. With this product, it's called Not Mayo. It's an eggless mayo. And we're Chileans. We're the 3rd biggest mayo consumers in the world per capita. And in only 5 months of sales, we became the 3rd biggest mayo brand. Not only that, but we conquered 5% of market share, achieved placement in 220 stores. And the best part of all is that our consumers are 85% non-vegans. So we're giving the same sensorial experience, or even better, with a lot of health benefits, with the same price as their regular mayonnaise, but actually the way we make it is more profitable than regular mayonnaise. So we want to scale this concept. This is our brand new 64 tons a month mayo facility. This will help us reach 500 stores nationwide, including Walmart, and generate a $4.5 million revenue a year. This is only by selling mayo. Our business model is simple. We direct sales to retail and food service, and we're licensing the access to our technology. Why are we better than the competition? Because this technology is allowing us to produce better products, faster, more cost-effective, and closer to the real thing than any other one. We're three founders: Karim Pichara, computer science PhD and research associate at Harvard, expert in machine learning; Pablo Zamora, biochemist, biotech doctorate from UC Davis, expert in plant genome, and he worked 8 years at the R&D department at Mars. Myself, I'm a finance major, foodie, and this is the third food-related startup that I created with one successful exit. We have a very nice team, and it's a mixture of experience and young scientific talent. The board of advisors has a lot of experience in the food industry as well, being Fernando El Solaro one of them, former Nestlé president for Latin America. The roadmap is clear. For 2018, we want to conquer Latin America. We want to scale the concept we have in Chile and bring it to Argentina, Brazil, Peru, and Colombia, generating revenues up to $2 million. Doing the same thing in the US, starting in 2019, generating in 2019 itself $7 million. We're recently raising $4 million to actually achieve this, and we're closing the round in the next few days. And this is the final slide. And tasting is believing. We have a whole booth for you guys to taste the future of food. Thank you so much.
Hi, I'm Mobhij, the co-founder of Deenalight Therapeutics, and we do gene therapy beyond the mucus. We are entering a new era of medicine, where your entire genome can be sequenced under $1,000, where cells can be taken from inside your body, outside, and reprogrammed to kill cancer, where animal genes can be edited to do whatever you would like them to do. Yet for diseases such as colon cancer and Crohn's disease, and the ones listed on here, the only options are to address the symptoms, such as inflammation, or terrible therapies, such as life-changing surgery or chemotherapy. These diseases are inherently genetic, or driven by genetic risks. So why is it that we are only managing the symptoms? As it turns out, that the cells that cause these diseases are hard to reach, because they're heavily protected by the mucus. Let's zoom into the colon to get a better idea of this. The cells shown in green are your intestinal stem cells. They replenish your entire intestinal lining every 4 days. So for any mutation in the intestine to be sustained, it has to occur in these cells. And they're heavily protected by the mucus, as you can see. In fact, the main job of the mucus is to protect your lining from the toxins and all the junk that you eat. In order for any biological therapy to exist and treat the cause, you must be able to get through the mucus and into those cells. When we started this company, we were really taken aback by one particular disease of the colon lining, familial adenomatous polyposis, FAP for short. A mutation in a single gene results in a 100% chance of colon cancer by the age of 40. There are no real drug options for these patients. So in their teens, they'll get their colon and rectum removed. They get an ileostomy bag. They still end up in the hospital because of nutritional deficiencies. And despite all of this, they still have an uncomfortably high chance of small intestinal cancer, which they have no options for. And we, in the 21st century, could not reconcile that the only option for these pediatric patients was to get an entire organ removed. And here's the kicker. We know that replacing this gene in these patients can reverse the disease entirely. But how do you really do that? Well, here's the colon lining of these patients. The stem cell is now shown in red. It has gotten a mutation that results in an overgrowth called a polyp. And that polyp will eventually turn into colon cancer. So again, For you to be able to create any gene therapy for this disease, you must be able to reach those stem cells and deliver the healthy copy of the gene. As it turns out, there are viruses, such as the poliovirus, which are able to penetrate the mucus and deliver biologics to the underlying cells. My co-founder and I came with a virology background, and we took our learnings from the poliovirus and translated them to a non-viral system. To be clear, we created a non-viral system that can penetrate the mucus and deliver cargo to the underlying cells so we can repeatedly dose these patients. Our vision for Deenalight Therapeutics is to replace surgeries in these patients, to replace chemotherapies for colon cancer, and to replace drugs in other diseases which only target the symptoms with a daily gene therapy pill that actually addresses the cause. In order to show that we can actually reach the intestinal stem cells, we took a fresh pig intestine, which very closely resembles the human intestine, and we dropped our delivery system on top of it, which is shown in red. And at 5 minutes, you can see that the vast majority of the delivery system is sitting on top of the mucosal layer. At 60 minutes, you start to see buildup at the bottom. And at 100 minutes, almost all of it is at the bottom of the intestines where the intestinal stem cells reside. We then took our delivery system with a fluorescent reporter and put it in animal models of FAP, which again very closely resemble the human condition. And you can see that in a tumor of those animal models, we were able to not only express the gene, but be able to get into the middle of the tumors, which have been very hard to do because of the mucosal barrier. We then took those same animal models and used the healthy copy of the gene. And we were able to show in just 4 weeks a 25% efficacy. This was one of the first gene therapy attempts for FAP, and it happened right here at IndieBio. We are now plugging this data into our platform, which is highly tunable, and to get to a clinical efficacy point that we desire. FAP is an orphan indication, which means that we're eligible for a breakthrough designation with the FDA, which speeds up the process. We are also eligible for a pediatric priority review voucher, which can be sold on the market for an average of $250 million. We expect to have our IND by mid to late 2019. FAP is our first indication, but it is not the only one. We are targeting a large variety of mucosal diseases. And the market sizes that you see on the screen are only for the GI tract and respiratory diseases. They don't even reflect other mucosal organs, such as the eye and cervix. FAP has a market size of $1.6 billion, just based upon replacing the cost of surgery. We are now targeting ileal Crohn's disease and MAP, another inherited form of colon cancer. Our IP is owned entirely by Deenalight, and we have provisionals protecting our ability to penetrate the mucus and get into the cells. And this technology is very tunable, and we can use methods to increase the efficiency as we desire. My name is Mubhij, and I did my master's in chemical engineering from Berkeley, and research at Caltech before that. My co-founder Tim is finishing his PhD in gene therapy at UC Berkeley. We're bringing together advisors who work closely with these patients as their clinicians and as patient advocates, who have created the animal models for FAP and other GI diseases, and who are pioneers in GI gene therapy. We have signed a term sheet for $1.25 million, which will let us optimize our first lead. At that point, we will raise our Series A, which will take us into humans. Deenalight is interested in talking to doctors, investors, and other parties who want to bring forth a new era of medicine. Thank you so much for your time.
Hi, my name is Kulika, co-founder and CEO of Shergill Logic, and we're recreating the best-kept secret in human breast milk. You all heard the saying, mothers know best, and it cannot be more true. There's something magical in human breast milk. For the first 6 months, babies can survive and thrive solely on it. It's their food, it's their medicine. evolution has tailored it just for our species. It's amazing when you look what's inside. You find lactose, lipid, and human milk sugars, which are even more abundant than the well-known hero, the protein. Human milk sugars are mostly unknown and unidentified until recently, but their importance hints by their abundance. 2-fucosyl lactose, or 2FL, is the most abundant human milk sugar. It's not meant directly for the babies, but for the good bacteria that lives in the baby's gut. So when a mother breastfeeds her child, she doesn't just feed one life, but multi-trillion that are essential foundations for the baby's health. But that's not it. They don't just feed the good bacteria, they fight off the bad ones. When treated with human milk sugar, a group of bad bacteria is basically broken down and dissolved, and this is something that would have caused invasive infection otherwise. Their benefits don't stop here. Beyond babies, in adult clinical studies, consumption of 2FL increases the abundance of the good gut bacteria by over 1,000-fold, an effect that not a lot of probiotic products can claim. Unfortunately, 2FL is found only in human breast milk, making it inaccessible to millions of babies who are not breastfed, and certainly beyond reach for the grown-ups. Until today, Shergill Logic offer the benefits of human breast milk without the breast milk. We recreate human milk sugars from scratch. Our product is identical to what's found in mother's milk. It is the next must-have ingredient in all infant formulas. We finally understand its function and importance, so there's no better time than now to bring the formula closer to what nature has to offer. Given its effectiveness in adult, it's also an ideal ingredient in prebiotic supplement and functional food and beverage products. It's fiercely competitive markets out there, and we can offer something unique. This is the very first time that adults can enjoy the medicinal benefits of breast milk. Our secret behind all this lies in something very tiny, yet tremendously powerful. We use yeast as a biocatalyst to make human milk sugar. Just like how beers are brewed, we take in byproduct from dairy processor along with simple sugar like glucose into our specialized yeast, a process that we have 4 IP positions around. Once brewing is done, we take our product out, separate, purify. Our final form is a powder. We're starting off with 2FL, but our technology delivers on varieties of human milk sugars. And you know what the best part is? It works. This is what we brew using our yeast. What I'm holding contains 2-fucosyl lactose identical to what's found in mother's milk. Our current cost of production is under $1,000 a kilogram, making us profitable today. This is where it's feasible for us to use it as an ingredient in infant formula, but as we scale, our cost decreases, opening up the opportunity in prebiotic supplement and functional food and beverage products, capturing the $25 billion market opportunities. For all these 3 market segments, we're approaching them through B2B sales. We're selling ingredient directly to businesses at 70% margin. This is not a 10 years dream. It's happening, and it's happening now. We already have $803,000 of booked revenues coming in sales next year. We signed 2 LOIs with infant formula manufacturer and adult consumer product provider. We also have a supplier lined up, signed LOI with one of the biggest dairy processor in the US. And we can offer what they're looking for because of our unique technology. We use yeast as a biocatalyst rather than bacteria or chemical synthesis. Our ingredient is the most affordable. Our process is regulatory friendly and consistently scalable, and we provide diversity of products. Speaking of regulatory, our product is approved and we're clear to ship our order next year. Since May, we engineer our yeast. We produce 2FL at bench top. We scale that up to 2-liter control fermenter, and we improve our strain effectiveness by over 100%. Our future milestones during the seed rounds are to further improve our yeast, move that up to pilot scale, manufacture at 10,000 liter, and ship out our first ton of ingredient to our customers. Over 80% of our production capacity is already booked. There's no better team than ours for these milestones. The three of us have known each other for 6 years during our PhDs at UC Berkeley. Our expertise are in pathway engineering, separation processes, and cellular regulation. We also have 3 amazing advisors who are never afraid to roll up their sleeves. Expert in protein design, pathway engineering, pathway design, co-inventor of the IP used in our work, XVP process design expert, each with 20+ years of experience. We're excited to open our seed round. We're raising $2 million, which will get us to our first $1 million in revenue. Our round is already firming up. There's no better time than now to unlock nature's best-kept secret. Join us.
Hi, I'm Robert Barkovich, CEO of Health Linkages, the data provenance company. We enable healthcare to trust and protect its data. The US, out of all the industrialized countries, the US has the most poorly performing healthcare system. In addition to be the most poorly performing, the US also has the highest cost system out of these industrialized countries. In 2014, 16.6% of the US gross domestic product was spent on healthcare. In 2015, that went up to 17.8%, or about 3.2 trillion US dollars. Unfortunately, this isn't expected to get any better. By 2025, the US is expected to spend almost 20% of its gross domestic product on healthcare, or 5.5 trillion US dollars. One of the biggest problems with healthcare right now is the data. People are hoping that the data will be used to reduce the cost of healthcare and to improve the performance of the healthcare system. But it's hard to do that with the data systems that we currently have. The US healthcare IT systems are antiquated. The core systems were built in the 1960s, and these systems were designed with an old paper-based workflow in which the data was designed to go into the system but not come out of the system. As a result, this ended up creating silos of information within each hospital and healthcare center, which were very difficult to access, particularly for the people who need to access them. The problem is, once you were able to find a way to get the data out, it still wasn't actionable, because since the systems were designed to take data in but not take it out, you ended up getting the value of the data without the context. And what do I mean by the context? Let's say you were able to extract a heart rate reading. You would want to know whether this heart rate reading came from someone who just went in for their annual physical or someone who recently had a heart attack. Hey, data scientists can eventually put this back together, but this isn't immediately actionable when it comes out of these systems, and it takes lots of man-hours to get it to that point. Fortunately, there is a solution. Health Linkages has designed a system based on a modern fast data architecture with a blockchain-inspired persistence layer that we specifically designed for handling and sorting large amounts of data, thus enabling the actors in healthcare to make better and more actionable decisions. We discussed blockchain, and this is part of our technology, but why is it actually needed? What does it actually do? Well, let me explain. The great thing about blockchain technology is that it allows people to trust the information. Let's take a look at these 2 readings. Imagine we have a blood glucose level and a temperature reading. What we would do is we would do a one-way encryption of this data called a hash, and that would be there in addition to the actual data points that we've acquired. We would then encrypt these data points together and encrypt them with the previous data entered, and so on, until every single data point is actually connected directly to every other data point. And what does this actually do for us? Well, let me explain. If we have any piece of data which is corrupted, which does happen fairly often, or that happens to have been tampered with, we can immediately detect this because, since I said every data point is connected to every other data point, everything's there. We're able to determine exactly when this happens, where on the chain it happens, and who the last person to touch the data was. Therefore, we get data integrity. We know that the data that we're using in the analysis is in the exact same state as it was when we collected it. We have data provenance, which is a complete event history of the data point, with every touch point associated with it. This gives us a quality measure of the data, which is essential for doing data analysis. Let's go through an example. This is Dr. Smith. Dr. Smith is the CIO of the J. Watson Clinic. Her board has told her that they're spending an extraordinary amount of money on diabetes patients, and they'd like to find a way to reduce the cost. She calls in Stanley Roberts, who is their lead data scientist at the J. Watson Clinic. Stanley realizes that they have Health Linkages core system, which maintains the integrity and provenance of the data, allows him to know that he's got access to every data point and every piece of metadata. So Stanley decides to begin his analysis. He takes a look at his data and he sees that within the past month, over 13,000 people were admitted to the hospital. He then sets his parameters to look for people who may be diabetic patients. He looks for blood glucose levels that are outside the normal ranges. He also looks for people who have been admitted to the hospital but haven't been treated for over 60 minutes after being admitted. He then finds 46 patients that fit this criteria. He then decides to chart this data based upon the treatment duration. Once they started the treatment, how long did it take to treat these patients? And he sees that all of these readings are actually on the very high side of the numbers there. They're all in the close to an hour's worth of treatment time, which leads him to believe that this is not strictly an insulin-related incident. There are complications that are being involved in this. As a result of this, Stanley's able to do an analysis. He's able to find that if he were to make some changes to the hospital policy and have these patients treated immediately upon coming into the hospital rather than waiting, they could save up to $2.76 million per year on these particular patients and their complications. In addition, since he was using Health Linkages system, Stanley was able to do this research in 10% of the time it would have taken him to do so otherwise, because the data was at his fingertips. He didn't have to massage the data and curate it to get it into that state. Health Linkages system is composed of two separate parts. The first part is called core. Core allows people to track the data as soon as it's admitted into the system, as soon as the data is collected. We create graph linkages to all of the different pieces of metadata, and then allow people to track the history of this data point through until its disposition, until it's finally disposed of, thus making the data much more actionable and allowing people to do analyses of it when they need to. The second part of our product is called Connect. And while Core works within a healthcare system, Connect allows different healthcare systems to share data for analyses with other systems without revealing their input data to the other institution. What this allows them to do is to maintain compliance with government regulations and also maintain the privacy of their own data assets. By doing this, it makes them much more likely to share data. I was talking to somebody from Kaiser the other day. He's like, why would I ever share my data? And he said, well, if I have this, I don't actually expose my data to anybody, but I can actually get some use out of somebody else's data and they can get use of mine, and I have no exposure. It makes perfect sense to him. The total addressable market in the United States is over 280 billion US dollars. It's growing at a compound annual growth rate of 15.9%, so growing very quickly. Health Linkages addressable market in the US is $2.8 billion and growing. Our business model revolves around license fees and professional services. The license fees vary depending upon the size of the organization and the services offered. We've calculated that our customer lifetime value is approximately $18 million per customer. We are delighted to announce that we have 3 ongoing pilots at this point, one with Temple University Hospital, one with UC San Diego, and lastly with the nonprofit group Open Cures. We are looking forward to continuing these and to having further deals with these institutions. We also have a number of other groups in our pipeline to be able to be pilot customers as well. Health Linkages is uniquely situated within the space. We built our blockchain-inspired system specifically to be able to handle large amounts of data and made it very, very scalable. It was also built specifically for enterprises, for private permission systems. We're also the only, to the best of our knowledge, the only company on the market that will allow people to share their data, share their granular data for analyses with other institutions without revealing their input data. Our team consists of myself, Nick Piscopio, who's our lead developer, Tinjun Chang, who's our architect, and Valerie Barnett, who's our counsel. Our advisors consist of Dr. Ken Mandel, Drew Rasmussen, John Wu, and David Snyder. Between the team and our advisors, we have a wealth of knowledge in the healthcare, life science, technology, and in particular, cryptography and blockchain technology space. In addition to that, Dr. Mandel is known as a pioneer in the use of health IT and big data to solve population health issues. We have our MVP developed. We have several pilots ongoing. We are looking to expand our team and our offering to be able to reach $1.8 million of annual recurring revenue by September of next year. As such, we are raising a round of $2.25 million. We are actually opening that round today, and we would love to talk to people about this. Thank you so much. I'm Robert Barkovich, and we are Health Linkages, enabling healthcare to trust and protect their data. Thank you.
Hello, my name is John Butler. I am the CEO and founder of QuantumSight. At QuantumSight, we're building tools and workflows for the single-cell biology research community. Now, why would you want to study single cells? Well, it turns out that the human body has about 70 trillion cells in it, and each cell plays a role in maintaining the overall health of an individual. So if you want to understand human biology and the human condition, and if you want to get to the core of disease, you've got to study individual cells. Now, the field of single-cell analysis is a new and emerging field, as evidenced by the increase in NIH funding for single-cell analysis studies in just the last 4 to 5 years. We're also seeing initiatives like the Human Cell Atlas, where the goal is to map every single cell in the human body. As an emerging market, researchers are looking for new capabilities to help advance their research. They're looking for the ability to take individual cells and place them into individual containers, but not just one cell, thousands of cells. They want the ability to treat those cells with reagents and then measure a response of those individual cells. They want the ability to go into the cell and pull out the genetic material, RNA and DNA, and they want to get sufficient quantities so that the data that they get after analyzing that material gives them strong conclusions. They also want the ability to associate that genetic data with specific cells in their test. And finally, they need flexibility, the ability to modify their experimental conditions in their lab without having to wait for a company to deliver a new product that may or may not help advance their research. Now, how does QuantumSight deliver these capabilities? Well, what we do is we put individual cells into drops on a piece of glass about the size of a microscope slide. We then take another microscope slide and deliver reagents into the same drops in the same grid pattern. We then touch the two drops in what we call a kiss transfer. Now, what happens when those two drops kiss is the cells respond to that reagent. And we measure how this cell responds and how this cell responds and how this cell responds. And we pull out, again, that genetic material, the RNA, analyze it on a standard sequencer like an Illumina or a PacBio, and take that data and figure out how those cells are responding and how the cells are responding differently. Now, if we can get this type of a product out, it's truly amazing what we can do to understand disease. We built a tool, we call it the Q1. The Q1 was designed and developed by our CTO and founder, Bidan Chowdhury. The system integrates some advanced optics from Intel that allow us to deliver the capabilities that we want to the customer. The system is housed in an environmentally controlled chamber, and the two slides I just mentioned are inserted into the system. The optics with the hardware and the software find one of the slides, they pick that slide up, and then they place it into a position directly above the receiving slide. You'd have the reagent slide and then the cell slide. The optics align these drops to ensure that we get proper and consistent delivery of reagents to those cells. And here's a movie showing the KISS transfer happening. One more thing I didn't mention is that we can actually address the cells, if you will, multiple times with different reagents. We have data showing that we can detect live and dead cells in our system, and we've shown that we can extract this RNA and measure gene expression in the live cells. We've also demonstrated that we can isolate over 65% of the total messenger RNA from the cells within our system. I'm proud to announce that in the last 4 months, we've booked $375,000 in revenue on the sale of our instruments. We have collaborations with Harvard, Stanford, as well as the Icahn School of Medicine. When we compare our technology to the current product families on the market for single-cell analysis for these capabilities, we show that the Quantum-Sight technology can deliver all of these capabilities, and we have a competitive price. The market for single-cell analysis is $1.6 billion with a 20% growth rate, and our business model is to sell instruments and consumables to the research market, and we'll be using third-party manufacturers to manufacture the product. Lifetime value of our products is about $640,000. We have 6 patents pending with priority dates of some of the patents back to September 2012. We've got a full-time commercially focused team with over 100 years of experience in high-tech, biotech, as well as investment. And we have a world-class science and business advisory board. Dr. Ron Davis from Stanford's on our board, Dr. Anders Granit-Jepsen, CTO of Intel, Dr. Chris Love from MIT. Dr. Chris Love is known for his expertise in the field of single-cell analysis. Dr. Carl Navina from Dana-Farber, Harvard, and Dr. Brock Siegel from Bioinflection Point Partners. He's not only a business advisor, but he is an investor. In the last quarter, we've booked 2 orders, and we'll be installing our first unit at Dana-Farber to start running clinical samples in October. We'll realize our first revenue in Q1 of 2018. At the end of Q1 of 2019, we'll be in production. We're currently raising $4 million. We have $1 million committed. My name is John Butler. I am the CEO and founder of Quantum-Sight. If you're interested in joining us in this journey, please contact me. Thank you.
Hi, I'm Mike Selden, CEO and co-founder of Finless Foods. Fish is one of the healthiest proteins we have available to us. It's high in omega-3, omega-6, and DHA. It should be what anybody reaches for when they're looking to improve their diet with something better. But lately, this hasn't been the case. Due to climate change and ocean pollution, fish has become something else. If you're getting your fish wild caught, you're going to have high levels of mercury and plastic in the food that you're eating. And if you're getting your fish from a farm, you're going to have high levels of antibiotics and high levels of growth hormone. And not only that, since farmed fish are generally fed fish that are wild caught, you're also going to have that mercury and also going to have that plastic. But despite all of this, demand is skyrocketing. It's doubled since the 1960s, and it shows no sign of stopping. If we want a return to the natural, healthy fish protein that we had previously, and we want to actually meet this demand, we're going to need to do something differently. At Finless Foods, we're doing something differently. We are growing fish meat, real fish meat, not vegan, not vegetarian, from stem cells. The way that we do this is we take a sample from a high-quality fish stock. We isolate just the cells that grow the fastest, grow them out in large quantities, and then differentiate them into the muscle, the meat that people like to eat, that muscle, fat, and connective tissue. From there, we structure it, package it, and ship it out. The initial species that we're working with is bluefin tuna. And this is not just a conservation effort, it's also a good business move. Bluefin tuna is an extremely high-value fish, and this technology is all about reducing costs. Cell culture already exists, 3D organ printing already exists. All we need to do is get underneath that extremely high price point of bluefin tuna, and we can get on the market very quickly. But the advantage of this is that we can get our price all of the way down until it's competitive with cheaper fish. We want to give people the option. Would you like to eat tuna, skipjack, and albacore caught with lots of mercury and plastic in it? Or would you rather eat the highest quality ahi toro from a bluefin without any contaminants for the same price? Our first products are surimi. This is a Japanese technology going back into antiquity, and it's extremely versatile. It's an extremely useful ingredient for soups, salads, you can fry it, it's a dessert, it's chips, it can be absolutely anything. And one of the advantages of working with surimi is that we don't have to reinvent the wheel in terms of structure. We can grow our cells out, make them grow extremely quickly and cheaply, and then ship them to people. Surimi science already exists. There are surimi scientists, there are surimi factories. We just need to supply them with this high-quality, contaminant-free material in order to get us to market faster. That said, our final goal is that same sashimi and fillets and steaks that people are used to. Our costs are lowering dramatically, and because of this, we'll be on the market 2 years after investment in restaurants, and then 2 years after that in grocery stores. Because of the nature of this technology, we're able to do something interesting with distribution as well. We can skip distributors altogether. Fish distribution currently is a massive web of middlemen. It's people on boats catching the fish, selling it to people at docks, selling it to people who are in distribution, and then selling that to grocery stores and eventually to the consumer. With Finless Foods, since our technology has no tie to the shoreline and no tie to fisheries, we can produce fish incredibly far inland, creating a logically distributed distribution system that actually not only kicks out these middlemen who are just adding costs on for the consumer, but also bringing fresh fish to areas where previously they had no access to it whatsoever. Our end user interest is extremely high. People want to create a better food system. People are interested in eating food that doesn't involve animal cruelty, that doesn't ruin the environment, and that is better for them. My co-founder and I, Brian Wyris, met 6 years ago at UMass Amherst, where we studied biochemistry and molecular biology, my focus being in epigenetics and his in primary cell culture. And we've built a team of people around us who are extremely qualified to tackle this problem. We have Dr. Robert Hughes, who specializes in biological scale-up. We have Dr. Ji-Hyun Kim, who is able to recapitulate the exact biochemical environment inside of a fish, outside of a fish. And then Torin Yeager, our tissue engineer. And our advisors are similarly qualified. We have Don Luning, the former CTO of New Harvest, one of the first people to map out how this industry needed to function in order to survive. We have Pavan Kambam from Clara Foods, who specializes in bioprocess engineering and bioreactor design, and Dr. Jay Park, who literally wrote the book on how to produce surimi. And because we have such a team of talented people, we've been able to accomplish a historical first. Just last week on Friday, it was the first time that anybody had ever eaten cultured fish. We are making history right now.
All right, you ready?
Yeah.
There you go.
It tasted like fish because it is actual fish. I did notice a couple things that were missing. It didn't seem to have mercury in it, and it was also missing the antibiotics that are rampant in aquaculture.
Why would you choose to farm or fish wild fish with all that environmental and human health costs when you could just have it made by Finless Foods?
So this is just one of many milestones that we've hit while being at IndieBio, and we'll be hitting many more very soon. We are currently raising a seed round that will accomplish two main goals. The first of which will be to create the first sashimi prototype that looks and has the same feel of the sushi that you're used to. And the next is to end our R&D phase. What this means that we will have all of the technology necessary in order to go to manufacturers and begin producing this product for sale in just 2 years. We are currently raising $3 million, and we will use that to take these cells that you are looking at and revolutionize the way that everybody eats seafood. I'm Mike Selden, and we are Finless Foods. If you're interested in helping us with this, please come and talk to me or my team afterwards. Thank you.
I'm Attila Haidu. I'm the CEO of Stelvio Therapeutics, and we're developing therapies through epigenetics. What is cancer? Cancer is uncontrolled growth of abnormal cells that don't die. This is what breast cancer looks like, and this is what brain cancer looks like. These are terrible diseases that impact all of our lives. So we started off by asking, if cancer is a developmental disease, can we reverse the process back into normal cells that can die of old age? Cancer stem cells divide symmetrically, which is basically a mother cell creating identical copies of itself. On the other hand, normal stem cells divide asymmetrically, which serves two purposes simultaneously. One is self-renewal, and two is producing generations of differentiated cells that can die. We tackled this problem using epigenetics because epigenetics controls gene expression. There's only two kinds of chromatin. There's open and there's closed. With open chromatin, the DNA is easy to read and the gene is expressed. With closed chromatin, the DNA is hard to read and the gene is not expressed. So we're labeling the histone methyl groups with antibodies, as shown in the orange shape in this diagram. We then invented a novel platform screening technology called microscopic imaging of the epigenetic landscape, or MEL for short. This is the nucleus of a cell. We've labeled the histone methyl groups with antibodies. Green is closed chromatin, red is open chromatin, and the blue is DNA. So we're segmenting the nucleus and extracting the features such as texture, bumpiness, and gradients. There are over 500 features in all, which gives us a pattern. We then use machine learning to recognize the patterns and assign every single cell a unique epigenetic signature, which is akin to a barcode. Now, the barcodes for a cancer stem cell are completely different than the barcodes for a normal cell. This is the core of our technology, it's cell identity. We can visually see the difference between a cancer cell and a normal cell. Next, you will see a new paradigm in drug screening with live imaging. Watch the cell in the center circle, and you'll see it dividing. This is important for two reasons. One, we can see asymmetric divisions, and two, we can track changes in the epigenetic landscape. Our approach is amenable to high-throughput screening of small molecule libraries. It's universal. It's applicable to many tumor types and other developmental diseases. It's 1,000 times more cost-effective than conventional sequencing. High speed. We can screen 1 million compounds in less than 1 year. But most importantly is the robustness of the epigenetic signatures, which can identify the hits, which is not feasible with other approaches. This is a distance map which takes the machine learning data from 500 dimensions and crunches it into 2 dimensions because that's all we need. So cancer stem cells map close, and normal differentiated cells map far, and here we can see which small molecules induce the reversion of glioblastoma cancer stem cells back to normal cells, as shown in the red and green dots, which map close to the positive controls of serum and BMP4. This is a distance map based on our gene expression data, which confirms our platform. We can see our top hits, in particular toposide, which induced the reversion of glioblastoma cancer stem cells back to normal. We're very excited that Johnson & Johnson have invited us to join J-Labs in San Diego starting September 26th. They're also open to using our platform technology to screen their compound library of 1 million compounds. We're also engaged in discussion with Sanofi to identify novel targets and therapeutics that could be used as treatments for neurodegenerative diseases such as Huntington's. So these conversations have opened up using our approach in other developmental diseases such as Alzheimer's. I've successfully executed our business model in my past many times, which involves partnerships with big pharma. We will use our technology to screen their libraries and come up with hits. We will then take these hits and optimize them through medchem, through structure-activity relationship work, and develop an understanding of the mechanism of action. We will then deliver these lead candidates to our pharma partners who will take them forward through clinical trials. In exchange, we'll receive upfront milestone and royalty payments. I'm Attila Heidi. I'm the CEO of Stelvio Therapeutics. I have over 20 years of experience in big pharma in all areas of the business, and I co-founded Stelvio Therapeutics with Alexei Tursky, who's our CSO. He's an associate professor of neuroscience at Sanford Burnham Medical Discovery Institute and an expert in epigenetics. We have a world-class team of advisors with David Andrews, an expert in machine learning, and David Spanner, who's an expert in the tumor microenvironment models. So why are we doing this? We're doing this to deliver medicines of value to the patients that need them. In order to do that, we're raising $4.1 million to operationalize our drug discovery and development efforts. Please join us after the presentations. We'd love to talk to you. Thank you for your attention.
Hello, everyone. We bring life to water and water to life. I am Kagiso Ubani, CEO of IndieBio. The world is growing at unprecedented rates never seen before. All this to the detriment of the environment. We continue to extract our everyday use products out of the belly of the earth, using millions upon millions of liters of water, using millions and liters of water to do this. To put this to perspective, to dig up a hole this big, you would need to take an entire river Ohio. This is water coming out of mining operations. It is highly toxic with heavy metals, highly acidic with toxin levels affecting decades to centuries. As this water continues to seep underground, it decimates and kills everything in its immediate vicinity. And this is a single example of a mine in Johannesburg. We have got tons and tons of such devastation worldwide. For such heavy-using industries, governments have set regulations to encourage safe and mindfulness use of water. Let's take, for example, sulfate limit discharge. 250 parts per million, it is potable water standard. Anything less than 1,000 parts per million, it is irrigation standard. Despite all these efforts, some companies continue to dump toxic water into natural waterways, and maybe they do so because the current solutions provided are way too expensive to implement or just not sustainable. However, companies need not fear anymore. We are providing a solution, a biological treatment. Now this is our baby, our heart. We built this with our own hands. This is in a place called Emalahleni. It is in the coal fields of South Africa. This is the heart and soul of our business. So this is what we do. The essence of what we do is this. we have found a way to enhance naturally occurring microorganisms, that way increasing its capacity to reduce toxin. And this platform has allowed us to convert toxic water into safe, usable water. Our process is multi-staged, and this is how it goes. Stage 1, using biology, we remove toxins. Stage 2, water is filtered using a filtration method of sorts as it's clarified. Stage 3, water is further cleaned and then is released for discharge. We provide our solution in such a format. This is a containerized modular plant which we can come into any operations that uses bulk water, plug the system on, and start cleaning water immediately. We're operating in a market that is growing significantly, and because we have proven concept of our technology, we are now confident that we can move into our secondary market, which is municipal waste, which is municipal sewage waste, industrial waste, irrigation, recycle, reuse, and seawater desalination. This is what separates us from our competition. As our competitors provide their technologies at 4 and 6 million, ours come in at 1 to 2 million. The byproduct of our process, it is a safe to use by human, valuable product. As prior said, we are running our first ever flagship in South Africa for Anglo-American. And this is a 300,000 contract where we're cleaning up to 250,000 liters of water a day. Additionally to that, we continue to have conversations with several other mining players, and these 3 are the ones that we are about to close deals with them. As a business-to-business company, this is how we make money. We provide our infrastructure at a once-off fee of $2 million. Additionally to that, we provide our bioreagent and our service at a cost. For investors, we stand to make $14 million out of a single mine if we look at the entire life mine span. We're building our patent portfolio as we protect our intellectual property, and as such, we have filed provisionals. Our milestones are as such. For the first 5 to 6 months, we intend to file patents, recruit staff, set up our facility, and much later on in our process, we intend to produce a single strain that has got the capability of reducing more than 1 toxin all at a go. I'd like to introduce you to a very exciting team where we boost experiences from water engineering, molecular biology, geochemistry, and I myself, I have got 10 years experience in molecular cell biology space. I've got 5 years experience as a laboratory manager, and I've got business qualification. Our advisory board is our nerve of strength. They continue to support us in our endeavors. We're raising $3.5 million. As we open this round, we would like to invite you in this journey of ours as we turn water to life and life to water. Thank you, ladies and gentlemen.
Hi everyone, I am Fatma Kaplan, the CEO of Pheronym. We use pheromones to control agricultural pests. This family was nearly killed by methyl bromide, which drifted into their house from a nearby application site. Methyl bromide is colorless, odorless, lethal pesticide. It is one of many toxic pesticides used in our food. Globally, $67 billion spent on toxic pesticides. They are making us sick. Consumers are demanding safe, eco-friendly pest control. Because of this, government regulations became stricter. They are being banned all over the world. However, to feed the growing world population, farmers have to control pests. They need safe, effective, eco-friendly alternatives. Nature already has a solution. Beneficial nematodes, which are microscopic worms, they search and attack insects. They're commercially available, they're safe, non-toxic, eco-friendly. However, commercial nematodes do not work as well as they could. Pheronym has a solution. I have been studying nematodes for more than 10 years. I identified the first mate-finding pheromone from C. elegans nematode. Then I discovered other nematode behaviors were also regulated by pheromones, both in beneficial and parasitic nematodes. This gives us the power to control nematodes. This is a video of dispersal behavior which is regulated by pheromones. The blob there was full of nematodes. Then we added the nematodes. Now these nematodes are searching for an insect. This is the technology we used to create our first product, Nemastim. It's a patented technology that provides safe and effective way to direct beneficial nematodes to search and attack insects. It's an additive to commercially available nematodes. We mix nematodes with Nemastim. Once they're activated, they're ready to spray to field. In the control, if you remember from the video, they were staying in one location. So the insect away from application site is still alive. The nematodes that treated with Nemastim, they were searching. They searched and found and killed the insect. Nemastim is broad spectrum. It controls commercially available nematodes that kill 28 most important agricultural pests. The addressable market is $12 billion. Furthermore, farmers can use already existing spraying equipment to spray the nematodes. So that means they don't need additional capital cost, and it makes it easy for our product to be adopted. In addition to Nemastim, we have 2 other products in the pipeline for a seed coat to protect the plants from plant parasitic nematodes. We are going to produce Nemastim synthetically because it is scalable. This bottle contains enough pheromone to treat nematodes for a 500 square foot area. We are going to enter the market from high-value specialty crops in greenhouses, then expand to fruit and nut orchards. We are going to sell Nemastim for $100 an acre. We are going to sell it directly to greenhouse growers and small farms, through distributors to access to 2.2 million farms in the US. We anticipate $40 million from direct sales and $1 billion through distributors. We already have pilot trials in soil with USDA, and greenhouse trials are scheduled. We have a letter of intent from Miraculex, which is a specialty greenhouse grower. We are establishing strategic partnerships for co-product development and global commercialization. Our main competition comes from toxic chemical pesticides. Our product is broad spectrum. Furthermore, our product is non-toxic and eco-friendly. our team uniquely suited to bring Nemastem to the market. I'm an expert in pheromone-controlled nematode behavior. My co-founder, Cameron Schiller, is an experienced entrepreneur with a background in economics. Our advisors and partners are Dr. Shapiro and Dr. Ed Lewis. They are leaders in beneficial nematode field and have helped other companies to bring nematode products to the market. They will lead our greenhouse and field trials. We are going to bring Nemastem next year to the market and scale up our production. In 2019, we will expand to international markets with Nemastem. In 2020, we will bring our second product, Ferrocode, to the market and increase its production. We are raising $2.5 million. We would like you to be our partners to bring 21st century crop protection to the world. Thank you.
Good afternoon, everyone. My name is Nicholas Pashers. I'm a founder and CEO of Bioaesthetics. At Bioaesthetics, we're focused on transforming reconstructive surgeries through regenerative medicine. And our inaugural product is a nipple graft. I'm sure we all know someone who's been through breast cancer or mastectomy. Probably most famously is Angelina Jolie, who had a preventive mastectomy a few years ago. But she's not the only one. In fact, there are more than 3 million breast cancer survivors currently living in the United States. Roughly 1 out of every 8 women will be diagnosed in their lifetime, leading to an annual market of $5 billion just on breast reconstructive surgeries from breast cancer. Now, after a woman has been diagnosed with breast cancer, she generally goes a mastectomy. A mastectomy is the part or complete removal of the breast tissue, including that of the nipple areola complex. Now, a nipple reconstruction actually offers significant health benefits beyond just aesthetics. Studies have shown that a woman is more likely to undergo a life-saving mastectomy if she can have a nipple reconstruction immediately afterwards. And that a nipple reconstruction actually increases self-esteem, body image, and decreases the feeling of distress a woman may have during a recovery period of a mastectomy. And current strategies to reconstruct a nipple are purely non-permanent and non-living solutions, such as prosthetic, rubber nipple areola complexes that you take on off at nighttime, non-living and non-permanent. Or you can have a tattoo of an image of a nipple onto the patient's chest, also non-living and non-permanent. Or there can be a reconstruction of pre-existing skin from the breast tissue, the thigh, or underneath the arm. However, these tend to lose structural integrity within about 3 years and lie flat against that patient's chest, leading to a non-permanent solution. Bioaesthetics' approach, however, is a complete regeneration, not just a reconstruction. And we start by using donor tissue, donor nipple areola complexes. And if you look at this underneath the microscope, all the cells and all of the DNA would show up as dark blue or purple. And the extracellular matrix, the protein, the stuff that gives it structure, the shape, show up as pink. Our technology allows us to remove all the cells and all of the DNA, all the blue stuff, but completely leave intact the pink stuff, the protein, the stuff that gives it its structure, leaving the outside aesthetics of the nipple completely intact. So we took this graft and we introduced patient stem cells, patient skin cells, and patient blood vessel cells to see how they interact if it was a safety profile. We saw a very low cell death and high cell viability. But as tissue engineers, we know that the biggest challenges are functional blood vessels and nerves innervation. We've been asked by plastic surgeons and patients alike, will there ever be sensation? And although that's a huge question that we can't answer right now, we can say that in a laboratory setting, we were able to grow nerves 111 micrometers deep just after 2 days of culture, showing the potential of this product in the future and where bioaesthetics is headed as well. Now, as for our functional blood vessels, for blood, we then engrafted this into a living model, into a mouse. Everything to the left of the blue line is bioaesthetics graft, and everything to the right is the host tissue. As we zoom in, we can see that blood vessel formation occurred, as indicated in blue, and new skin formed, as indicated in green, just after 3 weeks of implantation. Now, one of our largest accomplishments while being at IndieBio was actually a large animal study where we engrafted several of these nipples. Now, although this is still ongoing, we're going to give you a little sneak peek at the data just after 1 week. Now, everything to the top of the blue line is biosthetics graft, and everything below it is the host tissue. The tip of the nipple is indicated at the top of your screen. We saw that just after 1 week, cells migrated all the way from the host tissue, the blue line, all the way to the tip. And that indicated in the green arrows, new skin cell formation was occurring around the base of the nipple. When we zoom in, we can actually see where the host tissue, indicated in orange arrows, actually migrated into the new biosthetics graft, indicated in green arrows, creating a completely new skin formation along its way to a completely regenerated nipple areola complex. Now, the way that Biosthetics works as a company is that we partner with tissue banks and organ procurement facilities, and we process this tissue in-house with the cost of goods approximately $500 per nipple. We then sell these nipples for $3,000 at high-volume clinics, then we'll go to low-volume clinics, and then finally hospitals for a widespread adoption. We already have 60 patients lined up for our graft, and 5 early adopters, 5 plastic and reconstructive surgeons, already lined up as well, one of which is from a high-volume clinic that does 700 to 800 breast reconstructions per year in the US. We'll utilize these early adopters for out-of-pocket expense payers, then we'll apply for a insurance reimbursement code after 2 years so that we can go into those hospitals to have a widespread adoption to become the standard of care for nipple reconstruction around the US. We were the first group to identify and to publish about this strategy to reconstruct nipple areola complexes. And we have a nationalized patent for which we have an exclusive option to license through Tulane University, which covers a method to process, to derive the graft itself as a device, and also a very special way to engraft it to encourage skin growth and blood vessel formation. Now, as for regulatory, we fall through a very special pathway through the FDA called HCTP, which stands for Human-Derived Cell and Tissue Products. And because we fall through this pathway, we're purely a registration process and not an approval process. And because we're a registration process, we require no clinical data in order to get to market, allowing us to hit market within 12 to 18 months of today. Now, 56% of all breast reconstructions currently utilize material that have been through this pathway of the FDA, such as Alloderm and Flex HD. Before going to IndieBio, we had just finished our small animal study in mouse. While being in IndieBio, we started a pilot study in large animal and began to fill out our tissue supply, signing 3 contracts. From here, we'll finish our safety and efficacy trial, apply for our FDA registration, and be within market and revenue within 12 to 18 months. My name is Nicholas Pashos. I'm the founder and CEO of Biosthetics, co-inventor of the technology, a National Science Foundation fellow, and I have over 10 years' experience of tissue engineering and biomaterials. My business partner, Billy Heim, is the CEO of the company, an MBA. He's an experienced entrepreneur with 5 years' experience in biotech startups, as well as has worked in project management and fundraising. We have a truly phenomenal board made up of biotech executives such as Sabrina Johnson and Sandra Sufal, as well as 2 plastic surgeons, Scott Sullivan and Abigail Chafin, who together, their centers actually make up 1% of the breast reconstruction market, and Dr. Bruce Bonnell, the co-inventor of the technology. As for our funding strategy, we're currently in the middle of our seed round of $3 million, which we have 1/3 of it already accounted for. This will allow us to finish our safety and efficacy trials through the FDA and into revenue within 12 to 18 months of today. We're Biosthetics. Thank you very much.
All right, guys. What'd you think? I hope you had a good time. I hope you saw some great companies and some amazing new technologies. And I want a big round of applause for all the teams in the back. They've worked incredibly hard in the past 4 months getting all of this ready. I mean, obviously lots of sleepless nights, but a lot of real hard work and pain and went into this. And again, it doesn't stop today. This is not this is an arbitrary moment in time. All of these guys are going to be continuing to push super hard and build these technologies to change our lives. For that, I'm super grateful to them, and for that, I'm super excited for them. So yeah, thank you. Another thing I want to say is I want to thank this team up here. Everyone here puts just as much work, and energy into the teams in the back. It's not an easy thing to do this every 6 months and get all these companies going and bring you all in one place. So thank you all for that. I'd like to hand the mic over to the man of the hour, Sean O'Sullivan, behind SOSV.
Hi. So I just love what we do here so much. I want to tell you a very quick story. On Monday night, I was visiting a friend. He's my age. I'm 53. You can say I look younger than that. It's okay. But he's my age, and I visited him at his house, and he is dying of cancer, glioblastoma. He's in stage 4, and he has a few weeks to live. And this is life sciences. This is a really, really tough problem. And we all are facing these problems, not just with the friends and the people we know in our community, but my mother died of Alzheimer's. My son has autism. There's every single one of us on this stage that's touched by the challenges that we all face in life sciences. And so we are tackling, with this accelerator, with IndieBio, and with the work that all of the things that we try to do at SOSV, is to take really, really challenging and difficult problems and really tackle these difficult problems. And we need your support, and we need your investment in these companies who are tackling incredibly important issues for us as individuals, and for our society, and for our future as human beings on the planet. So I ask you, to reach deeply into your hearts and into your minds and look at these companies in depth and figure out if these are companies you really want to support and invest in and help them on their next stages of their journey. Because this is not another app. This is not an e-commerce play that may go for a year or two and blow up. These are things that are actually going to affect us and affect our lives for all of us. So thank you very much for coming today, and thank you for supporting these companies. And I'll turn it back over to Arvind for some logistics and for some thoughts. Thank you.
Thank you, Sean. So again, thank you all for coming. It's a real pleasure and an honor to have you. But it's not over. Please go make your way out to the foyer and where you're welcome to meet with the teams, chat with them, get to know them if you haven't met them already. And again, one thing, it's almost all investors here anyway, but just in case, if you'd like to meet a team and they're talking to investors, very rarely do these teams get to see so many at once. So please kind of hang back for the first half an hour or so and let them get through those conversations. That would be appreciated. So thanks again, everyone, for coming, and we look forward to seeing you in about 6 months from now for batch 6. Thank you. Bye.